built over borders and things like that. And when we send data to the control programs, we send these sort of big spreadsheets with genotypes and so on, which, of course, by themselves are not really very useful until you can do something like map them out. So we got an idea recently for a tool that would allow you to look at this data in a somewhat different way. We have in the presentation you saw earlier, you've seen that we've identified clusters, clonal clusters, and connected them and so on. And what we thought is, actually, this should have some sort of timeline. Did this cluster appear before the other cluster? Did it move across and so on? So we've developed a tool that actually can show you how the data that we deliver has evolved over time and over space. And you can actually sort of look at how these clusters, how these strains flowed and move across the territory. So this tool is called EpiGenTracker, and I'll leave you with Varanya, who's the mastermind behind this.
Hi everyone, yes, my name is Varanya. I am very pleased to introduce to you a new tool that the GenRe-Mekong project has developed. It is called the EpiGenTracker, and it is a browser-based tool that for exploring data through time and space. With just a few clicks, you can turn your CSV data and into putting them directly into the maps. And the map is moving, it's dynamic, and it gives you timeline, and you can see things on the charts. So you can see how the samples changes over time. You can compare regions side by side. And if needed, you can also visualize the genetic relatedness as well. So why are we developing another tool? Well, surveillance data are complex. There's a lot of information coming in from different places, different time, and it can be difficult to get a clear picture of what is actually happening. The GenRe-Mekong project has already developed a tool that helps translate surveillance data into actionable public health information. Like specifically, it takes information from the spot malaria genetic report cards, and it generates intuitive geographical maps of prevalence, diversity, and relatedness. But with the amount of data to mine for different use cases, we need a collection of tools to help answer different questions. There are already many existing tools for exploring genetic data, but many of these can be quite complex to use. They're quite technical, and maybe they're not that suitable for routine program settings. But EpiGenTracker was created to make this easier. It is a free, user-friendly tool that focuses specifically on genetic epidemiology use cases, and it works directly in your browser. And most importantly, it helps translate real questions into insights, like questions like how many cases are we seeing in each area, where are particular strain appearing, where are they spreading, and what has changed from last month or last quarter to the next, for example. And with this tool, you can just use your mouse to click on the timeline, and then you can drag along the timeline, and you can see your samples appearing on the map for that particular date or time period. And this is the main difference between this tool and the previous tools. So the idea is to make it simple for you to explore your data quickly and see the patterns that can support decision making. So let me briefly walk through the main features of this application. So you can move through time and geography to see when and where samples appear. The markers, each of the dots are one sample. So you can color the dots, your sample, by any grouping of your choice. For example, you may choose to color the dots by species, by artemisinin resistance, by cluster number, or anything really, as long as you have a column in your data sheet, you can give them a color. And so then you can also explore the temporal trend and then summarize the data in the chart at different administrative levels. It can be at district level, province level, and country. And the color also correspond the same color as your map. So if you set it as, let's say, species, for red is PF and blue is PV, for example, then you also see that on the chart as well at the same time. And if you load the a distance matrix, the tool can display an interactive genetic tree, and this can give you a visual sense of how the clusters are related to each other. And finally, by combining the maps, timelines, and the color, the grouping for your color, you can identify meaningful patterns like the emerging strains, for example, outbreak hotspots or cross-border spreads, or any unusual changes from 1 month or 1 quarter to the next. Okay, so what data does it need? There are 2 main parts. It needs a map and it needs your sample data. Currently, for the map, there are 2 options. You can already load up the map of the Eastern GMS that has 4 countries of Thailand, Cambodia, Vietnam, and Laos. And if you have your data sets for these countries, you put your data set in, then it's already plotted on the map. Or you can use your own map. So no matter where you are, where your samples are from, you can supply a map which can be downloaded from this database called the GADM database. It has the map from every country in the world. So in some way, this application is limited to just exploring data just within this region. And for example, the minimum requirements is you only need 2 columns, 2 things. One is you need a date, and the date can be the full date or just the month and year. So we can just plot with month and year as well, or the full date. And the other thing is the administrative division 2 identifier, or some country called district identifier. And this identifier is the official identifier that you can get also from the same website, the GADM website. For example, this is my example of a data sheet that I can supply. Actually, I don't need any other columns. I only just need the orange column that says the GADM ID for my district and the collection date. For example, in Massot, the ID this is the ID, and all of this can be found from the same database that you can get your map, because they have the information for the administrative division to administrative divisions, different levels for every country. But in your data sheet, if you have other columns, for example, like a species or latitude and longitude, it just makes your map look nicer, because if you supply latitude and longitude, it puts exactly where it is. But if you don't give it latitude, longitude, then it puts it in the center of your district, for example. And other columns, if you give it to it, like the species or like country or anything, you can choose to color your sample by that column. So it can make your map a bit more meaningful. And if you are already using the genetic report cards that GenRe-Mekong give back to our partners or that we have published, you can already just convert them into CSV file and upload directly. Already can explore your data, throw it in, and you can see your data already appearing, which we will do in this session later on. And importantly, data never leaves your machine. Everything runs inside your browser, and nothing is stored, nothing is sent anywhere. To your data, we do not have access to your data. Okay, so now that you have seen what EpiGenTracker is designed for and the kind of data it needs, we are going to move into the practical part of the session. We will begin with the live demonstration where I will walk you through the application using an example use case of tracking the PV strain over time. And so for this part of the demonstration, I would like to ask everyone to really pay attention to my screen before already doing things because it's a lot of information. And then after that, you do have a chance to explore the tool yourself, where you get to explore the tool using the already published PF surveillance data, and that I've sent to you in the email, or if you do not have it, we can give it to you. So yeah, you get about half an hour to explore the data yourself afterwards. And before we start the live demonstration, I do want to take a moment to acknowledge all of our partners who made this PV surveillance data possible. So a big thank you to all of our control program partners and scientific partners as well. And would like to thank our funders for supporting the GenRe-Mekong project. So the data sets that I will use, you have already seen the results from Wipari's presentation this morning, but now you get to see it in a bit of a different way. So to access the data, we go to the GenRe-Mekong project website, and I am going to need to, yep, GenRe-Mekong, not that. So I think all of that, as long as the GADM website they updates, right? You get the yes, for the practice. So when you go to the GenRe-Mekong website, to access all of our tools, there's a tab that says tools, and in here you can see all of the tools that we have developed and will develop. But for now, we also have a shortcut to the EpiGenTracker, as you can see at the top, or it's in the tool page. So when you click the EpiGenTracker button, it takes you to a page that will contain all the tutorials, user guide for this application, which will be coming soon. So, but to launch the application, you just press the yellow button, launch application. And straight away, you get a welcome page. And if you have never used this application before, you can have a tutorial, like a quick tutorial, start tutorial, and it would highlight the different parts of the application where you can, where you interact with. So on the top left, you can load your data up here. And at the bottom is where there's the timeline that you can drag and move through your date. And when you update when you go through the date, the map will update as you go along, as well as the shots. On the right-hand side, you get the shots, and you can choose how you want to see the data, which you get to see all of this in a bit when I started to demonstrate. And you can also the color for your dots for your sample. Just below that, it's the legend. There's also an option to upload matrix for your cluster, so you can see the genetic similarity tree for your cluster. Okay, and then we're gonna click Start Exploring. So the first thing that you have to do is to choose a map. So we're gonna load the Eastern GMS map. Oh, the internet's fast. Okay, so straight away we get the map, and I am going to load my PV surveillance data. It's in a CSV file. And I'm going to stop. So when I first loaded up my data, straight away what you see is things already appearing on my map. And at the very top of my application you see the date. And if I move my timeline down here, you see that the date changes and my map get updated. So just to not make this confusing for now, I'm just going to color it by country. We will come and talk about the actual data afterwards. First, let's focus on a little bit about the application, like the things on the screen, so that it's not confusing. So each of these dots is one sample. It's one row in your data sheet. And my dot right now is colored by country. And so that's why you get to see the same country, same color. And yes, so you have seen how if I drag, things updated on the map. You can zoom in and out. Yep. And then let's say for the shots, right now I can choose to do it by country, and I can just click. And right now there's 4 options because in my data sheet, I only have 4 countries. So it will not show you anything you don't have. So I can just click and select, or I can type as well. I can just type and then select. This application is a little bit better when you have a bigger screen. Okay. And so this is my chart. The first thing I wanted to point is the y-axis is the number of samples. The x-axis right now is showing the month by month, and it's showing just 3. Okay. And let's say we move through the time, you can see that the chart, updates as we go through the time, just like in the map. So this can be something that is helpful. We can also look at it at district and province level as well, not just the country. And we can also aggregate these data by quarter rather than by month. So if you click it here, then it changes to quarter rather than by month. And if you don't want to just see the axis to align, so right now it's aligned. So when I move the time, both of my charts move at the same time. If I don't want to see that, but I want to see all the data, I can unclick this. And then when I move along, then I'll see all the data for my and it may not be aligned. For example, here you get to see all the data. Okay, so then next, I just wanted to point out, but may not explain this part, which is the control on the left-hand side. This is where you can make your map changes a little bit of how it looks. Each of these steps, each of these control you can read about them a bit more in this help button. Like, for example, we have an option that move forward in time. Like right now, it's moving forward in time, 7 days in time. So the next day, okay. So the next day that if I allow the animation to play, that you will see for this one, it will add 7 more days. So it will be 12, for example. It's because I've set at 12. But all of these controls, just to let you know that the default is already okay, already good, like we already use this. You can also play around with it. You can change the animation speed or how long you want the dots to remain, like the lifespan of the sample. For example, right now we are showing actually the dot to remain 60 days. So if I change it to only 1 day, then only the sample that for this date will appear, which is none of them. Oh, just a few, yeah. So you will only see the sample for just that date, but then it would be not as easy to visualize, I think. But the setting is really up to you. But the default is already okay. You don't have to play around with it at the beginning if you want. So now I am going to actually show you the use case of PV, tracking the PV strain. Right. So I'm going to change this to colored my samples by cluster. So straight away, you see two things. You see that my map becomes a lot more colorful, and my chart also updated to be a bit more colorful, as well as a lot of gray. And this is because, so if you remembered from our presentation, for our PV surveillance data, we use the microhaplotype to group together parasites that are similar with each other. And so we group together parasites that are similar at 90%. So each of these color pretty much represent clonal cluster, so it's represent one strain. So if the sample is colored by the same color, then it's the same cluster. And so we have identified in our PV surveillance data set 47 clusters, and the samples that are not part of any clusters are in gray and given a triangle. And so with so many strains, we're not going to be able to track them all. So we're just going to track the one that is most interesting for us at the moment, which I am interested in seeing strains that might be very similar to each other. And so for that, I will give it the genetic tree, which, the genetic, to draw the genetic tree. We have the genetic distance that we compare how similar each of these clusters are, and then we just use it for this application, and it will summarize it for you in a table. In the future, GenRe-Mekong do plan to give this distance matrix for the cluster to our partners as well, so that you can just also explore these data yourself. And this table, actually, it's a long table, but the nice thing is it's arranged by the highest genetic similarity at the top. So the one that I'm interested in right now would be, so these two, we have two pairs of clusters that are similar to each other at 80%. So very highly similar. But I'm going to be tracking this one that is cluster 4 and cluster 6, because they are very large cluster and they are very similar to each other. So to track them, I can press onto the color 4 and 6, and what you will see is that things changes a little bit on your map and on your chart. First of all, the color disappeared for the labels that you did not select, and for the labels that you select, it will be highlighted. So I think because our cluster is light in color, I'm going to change the theme a little bit. It'll be easier. Like chart, you see that because I only select 4 and 6, my chart is, everything else is faded away, only the yellow one get highlighted. Okay, and so now let's maybe start tracking these two strains, which we know from our analysis that this morning that they circulate around this area, which I will zoom in, and let's start at the beginning. We can also, like, I'm gonna show the province label so that we know where things are. So here, okay, so I'm just gonna drag so that it's easier. So my cluster 6, yellow one, first appeared in this district in Quang in 2019. And we see that it is found pretty much next, along the border, at the collection site, very next to the border between Laos and Vietnam. And so it's kind of circulated for a bit. It goes to the other district, in Quang Tri, as well other districts, but still within the same province. Right, and so, and then at the end of 2019, we see that the first sample of this yellow strain, cluster 6, crossed over to Laos, to Savannakhet, Salawan, to Samui district in 2019. And so it keeps circulating in Salawan, and we see a lot more of them in Quang Tri. And then in 2022, it crossed the border again, but now to another province, into the Savannakhet Nong district. But if we notice, we see that the collection sites that identified these clusters are actually pretty much along the border, very next to the border. Right. It did move inwards a little bit in the same district in Nong. And then suddenly in 2024, we see this rapid increase in cases that was identified as cluster 4. So this was an outbreak that we have seen and heard quite a lot today. And this outbreak in Samui. This outbreak in Samui was, from our data sets, showed that it was caused by the same strain, cluster 4. Same strain, the cluster 4, which when we look at the genetics, we see that they are 80% similar to the cluster 6, which has been circulating for a long time. So let me actually give you a bigger picture. You'll be able to see that. So the cluster, you can see that the yellow cluster 6, has been circulating around for a while. And then suddenly, like the outbreak in Salawan in 2024 contributed to most of the samples collected in Laos. And this data set shows us that PV strain can circulate at low frequency for a long time. It's very persistent. You can see that the yellow is low in number, but it's keep popping up for a long time. And then eventually it's recombined with other parasites to form this new strain that seems to be doing better than other parasites around, and is causing this outbreak. Yep. And so I hope that through this use case, you get to see a little bit of what application can do. And so, yeah, now it is your turn to try out the application. And if you have any questions, feel free to ask, put your hands up, because we do have a lot of helpers to come and help you. So, so in order to access the application, you can go to the GenRe-Mekong website. Yeah. Oh, does anyone have any questions? Yes.
Thank you very much for introducing a very useful and interesting tool. And can you explain more on the data set about the formats of all the variables? Is there any specific requirements, like the name of variables, or it automatically recognize it as a level 1, level 2? And can we add other information to other levels of categories to that or not?
Yes, you can. Sorry, I didn't include too many information, but I'm very happy to work with you and show you all the requirements. But the most basic requirements is what I told you, it's just 2 columns, it's the date and the ID for the district. But if you supply the name, the actual name, write it out. If you say, let's say, Administrative Division 1 as the header, and then you give it name, then this application will use your name onto the map rather than the name given by GADM, because in this region we do have maybe different ways to spell the names of the area. And so if you have a particular way you want to spell it, you can put that column in and it will use your name. Yeah.
Yeah, for example, for the data in the data set that you sent us, I downloaded and I see that one column is for year and one column is for month.
Yeah.
Can we put it in together?
No, you can already put it in. So if you don't have the actual day, right, and you have a column that says month and year, the program recognizes it already and will use it
separately.
Yeah. It's the reason why the data set that we shared with you is the one that we have already published, and with the published data, we don't normally give the day. So that's why this application can also just use the month and year to plot. Yeah.
And do you have the option of showing cumulative data instead of for each date or month? I mean,
like cumulative for
a period of time, for a specific period of time. Like in 2025 in one map, in 2024 in one map, not by
target period. At the moment, we do not, but actually, I can show you maybe one of the ways you can play around with this application, how you can just show all the samples, maybe from 1 year on the map. We do plan to improve this application because it's still very, very new, like almost like a prototype that you can play around with already, to export, save as like some kind of a report that you can specify the period that you're interested in. Yeah.
Thank you very much.
Other questions on this so far?
Okay. Yeah. Feel free to during this time. If you need a little bit of break, you can as well. But feel free to try it out yourself and just make sure you have an internet. Go to the GenRe-Mekong website.
Can you just remind them what the data set is?
Yes. So the data set that I think I have sent to everyone, I've emailed you last week, is the PF surveillance data from 3 countries that we partner. It's a surveillance data from Vietnam, Cambodia, and Laos. These are the samples that has been collected by our control program partners. And we have published this data earlier this year in the paper that I think I also sent the reference in the email. Yeah. And the very nice thing about this data set that you will get to play is we have drug-resistant information. So you can see those like a drug resistance because it's PF. So you can on your map you can see the red and green of resistant and sensitive, resistant for the drug. And then you can, if you put like a chart as well, then you get to see the changes in time and different location. Okay, feel free to, if you need help, please raise your hand. We do have quite a few of our GenRe team to come and help support you if you're not sure where to start. Yeah.